Okay, let's talk about these modifications of genetic ratios. Here we go. So ABO blood type is not going to have the same ratio. Probabilities in the ABO blood type and albinoism. So in this case, you have a lethal allele. So this is exactly what I was doing. So of all the offspring, 3/4 are pigmented, 1/4 is albino. Okay, do we agree with that? Then of the pigmented, of all the offspring, 3/4 are pigmented type A, 6/16 are pigmented type B, and 3/16 are pigmented type B. Then you have albino type A's, albino type ABs, and albino type Bs. Does that make sense? So it's a little bit of a weird ratio because you have the albino in there. Okay. Gene interactions. Sometimes they're not necessarily due to an interaction of genes. They just, red hair and green eyes sometimes tend to go together. Cellular function of numerous genes contribute to the development of common phenotypes. Epigenetics. Epigenesis, eye formation, shape, text, and color. Each step of development increases complexity of the organ under control of one or more genes. Deafness, mutation interrupts step in ear development, influenced by many genes, multiple interactions. Epistasis, expression of one gene or gene pair masks another. Example, the Bombay blood type. Doesn't matter what your real blood type is. If you have this Bombay blood type, it's epistatic. It means it masks the other ones. It's epistatic to other ones. This is the Bombay blood type, which we already talked about. Gene interaction sometimes reveals an inheritance pattern that's modified to the 9:33:1. If you have epistasis, you'll see just a weird ratio here. Inheritance is not as continuous as you would like to be. And the genes are not linked. We'll talk about linked genes. We're not doing it yet. So you will just get a very strange ratio here. Instead of 9:33:1, you'll get a 9:3:4, a 12:3:1. You'll just get some of these ratios that are off because you have a modified situation. Here's mice coat color. We did one of these in class. Black pigment, agouti pigment, all black, no black pigment, even if A's are present. So if you add this to the mouse when we did in class, if you have this BB genotype, you have no pigment at all. You are an albino mouse. So if you look at that, mice have this same thing with the albino. Squash do it with white color. Peas do it with purple and white. Squash, chicken color, mice color. Shepherd's purse is a type of plant, and flower beetle. They all have these kind of strange ratios. Dominant epistasis. Dominant allele masks an allele at the second. So summer squash fruit color. The dominant allele is white fruit. So if you have this dominant white fruit, it doesn't matter what your second allele is. You're getting a white fruit. So any absence of this A allele, anytime you don't have an A allele, such as these options, little a, B, B, then you have a yellow fruit. And if you have little b, you have a green fruit. So can you see this is going to be a weird genetic ratio? We may have to do one of those in class. But if you just think through it, you're like, okay, this is dominant to that. A is dominant to everything, and you get a white fruit. If you have any A in the equation. So that is going to look like this. You do the third generation. Complementary gene interactions. These are crossing of two breeding strains of white flowered pea plants. You get two true breeding strains of white sweet peas. F1, all purple. F2, purple, 7, white. This is just a complementary gene interaction where they complement one another, and at least one dominant allele for each loci needed for purple flowers ensures proper biochemical conversion. Same as the white and the yellow. So a disc-shaped fruit crossed with long fruit, all disc-shaped fruit, and then you have 9:3:1. And this is because you have the influence here of if you have any A and any B, it's a disk. If you have any A and little b, it's a sphere. But also if you have any little a and a big B, it's a sphere. This is a weird ratio. This is like something where you'd have to really read the problem to see that this is happening. And then the totally recessive is just long. Do you see what we got there? Totally homozygous is disk. Totally homozygous dominant is disk. Totally homozygous recessive is long. And everything else in the middle is a sphere. Here's what fruit looks like. Disc fruit required dominant alleles at both loci. The sphere fruit on either. The long fruit, no. So here we have the sphere, and it is basically, oh, which one was the sphere? I gotta go back. Heterozygous. So long fruit, go back up here. Long fruit, no dominant alleles. Sorry. Long fruit, homozygous recessive, right? If your fruit is long, you are homozygous recessive. If you are a disc, you are homozygous dominant. And then your spheres are the recessive, are the heterozygous. Heterozygous. So these guys, spheres, heterozygous. Disc, homozygous dominant. Long, homozygous recessive. It's really not, it's just one of those things where you have to read the question, and it'll say something like this. I don't expect you to know that. The principle of segregation and dependent assortment makes these things happen. That's Mendelian ratios. Complementation analysis determines if two mutations are on the same gene. So we will do gene linkage and look for the gene order. We will do that after the next test. But I do want you to know that there is such a thing as the genes being linked because they are on the same chromosome. That happens. So here you have a complementation analysis looking at two wingless mutations of Drosophila. So here you have a homolog on here and on here, and here, right, you have a complementation. Here you have no complementation. So we will talk about that more. There's a whole chapter. But I just want you to know that it is possible for you to have, if the alleles are on the same gene, they're going to potentially sort that way, and it's going to throw off your Mendelian ratios. Pleiotropy is something that has multiple phenotypic effects. Really, lots of things have multiple effects. Like anything that has a range of colors, range of disease, it's pleiotropic. So Marfan syndrome, autosomal dominant mutation in connective tissues in the fibrillin results in multiple phenotypic effects, affects the eyes, the bones, and other tissues, but it can be like really mild or really severe. Another example of there's some autosomal disorders. Inadequate metabolism of porphyrin results in toxic buildup in the system. Anywhere from just abdominal pain to fever to racing pulse to insomnia to vision issues. There's just a range of effects. X linkage, we talked about this. It causes a unique pattern of inheritance. Typically it's in Drosophila, but it's really in everything. So if we did it in Drosophila, it would look a little bit like this. I use the X and the Y. I don't use these because I never remember them. Genes present on an X chromosome exhibit a pattern in which males tend to have more, and you would see that in any kind of, when you look at a history of the, what is the word I'm looking for? Pedigree. When you look at a pedigree, you will see that mostly the males have it. Males cannot be homozygous or heterozygous, but the word there is hemizygous. And so you will just see, again, there's a lot of them. Color blindness is one. Here, right, the mom has it, and then all of her sons have it, and this daughter is a carrier, so her son over here has it. And if you can't see this 15, you're probably colorblind. You need to come talk to me. There are some lethal X-linked recessive disorders that